That brings us to our last speaker, my former boss, Will Bohr, who will probably tell us something about DNA repair, I'm thinking. Thank you so much.
Well, thank you, Morten. It's really a pleasure to be at this meeting. It's very exciting, and it's also nice to see your very good work coming out now. So I'm going to talk about some of your old work and some new things here. So let me start with that the DNA damage response is central to the thinking in our lab, and the DNA damage accumulates, as we know, over time, and so importantly, it can be specific lesions in DNA, or it could be DNA alterations. And what's appearing more and more is that it doesn't have to be a very well-defined DNA damage, but it could be a change in DNA structure that causes a signaling process. So it can be repaired by DNA repair, which is our particular interest, or if it's left in the genome, it causes a large number of changes and pathway inductions. And so it's a very broad and profound DNA signaling thing. And you can have various immune responses, chromatin structural changes, transcriptional changes, replication changes, and then also, in addition, the work that we've been doing and others suggest that there is a pathway through mitochondrial deficit, mitochondrial damage, that probably involves the phosphorylation process. And then this damage to the mitochondria causes mitophagy, bioenergetic problems, and could be very important in senescence, aging, neurodegeneration, and also cancer as well. Now, the phosphorylation process we've heard about at the meeting, and it's a very central process in the DNA damage response. And there are many PARPs, but PARP 1 is very functional in this. It's involved in the DNA repair process and the phosphorylation process, which uses NAD as a substrate. And interestingly, PARP phosphorylation with PARP 1 occurs many places in the genome, and I find it very challenging and interesting to understand where this signaling particularly occurs. In our lab, we have recently worked on mitochondrial phosphorylation, and we have some new work coming out suggesting that there in fact is phosphorylation occurring inside mitochondria, as well as we know from other studies it occurs in the nucleus and other regions of the cell. So with this phosphorylation, we deplete NAD pools, and therefore all other enzymatic processes that depend on NAD, including sirtuins and many others, and that can then be part of the process towards mitochondrial dysfunction. So the work that we have done over many years has pointed out that there is a particular phenotype associated with deficient DNA repair, and mainly in the diseases that have marked neurodegeneration, which is often seen in DNA repair diseases. So we started with these disorders here, and as you can see, many of the papers here have Morten's name on them, and Morten was instrumental in the early process of identifying this mitochondrial dysfunction in these diseases of DNA repair deficiency and neurodegeneration. And we used various approaches. We used cell lines, mouse models, nematodes, and bioinformatic approaches to determine this mitochondrial dysfunction, which was in many cases mitophagy. And so the phenotype in these diseases were increased phosphorylation, increased mitochondrial ROS formation, membrane potential issues, and then lower NAD levels, probably in accord with this hyperphosphorylation. Decreased sirtuin activities, and also that these diseases had a mitochondrial phenotype, which was noted in Morten's database at the time. We have since found that this phenotype is also seen in other DNA repair deficient disorders, Werner syndrome and apotaxia. Apotaxia has neurodegeneration. Werner syndrome may or may not have neurodegeneration. And then in Alzheimer's disease, it's very prominent as well. And we spent a lot of effort on Alzheimer's in recent years to understand this process. So the model that we have generated has to do with this. There's a lowering of DNA repair is seen in these diseases, it's seen in Alzheimer's, and it's probably also, well, it's known that DNA repair declines with aging, so part of the aging phenotype could also be this mitochondrial dysfunction. We have DNA damage accumulating in the nucleus, also in the mitochondria, but because of various findings, we think that it's a nuclear damage that's particularly significant in this pathway here. And it is in part because some of the proteins that we have deficiencies are in the mitochondria and others are not, but they have the same phenotype. And then we see this pyrrolation increase, depletion of NAD, of sirtuins, mitochondrial dysfunction, and several kinds of mitochondrial health problems, notably mitophagy, which we have particularly been interested in. And we have then used various interventions, mainly NAD supplementation with nicotinamide riboside and NMN, but also urolithin A, which we have studied we screened for mitophagy stimulation in Alzheimer's, and we found that urolithin A was a very potent stimulator of mitophagy. So now we're comparing pathways and features on behavior and also various other bioinformatics on the stimulation with urolithin A versus nicotinamide riboside approaches. And with this, in these models with NAD supplementation, we see a lot of improvement of these features that I mentioned before. And also NAD has a direct effect on DNA repair. So it has a mitochondrial stimulation, mitophagy, mitochondrial health, but it also improves the DNA repair function, which is of interest to us. This work has then led to that there are a couple of ongoing clinical trials. We have in Copenhagen syndrome and in Werner syndrome, and at this point we don't have any results to show, but it is ongoing as orphan disease intervention. So I'll talk a little about some findings in Copenhagen syndrome, and that was already mentioned this morning by Morten. It's a very interesting rare disease, which has a very severe aging phenotype. It's a premature aging. Patients have very severe neurodegeneration, a lot of other features, and there are two complementation groups, CSBA and CSA and CSB, and those are different genes with different functions, but yet the patients have similar phenotypes. So if you want to understand the real phenotype behind Copenhagen's, you need to look at both of these genotypes and what are common in terms of the pathways that lead to the disease. And importantly, interestingly, they have severe transcription problems, DNA repair problems, but no increased cancer. And here is a series of two important clinical studies on the features seen in the Copenhagen's patients. And very importantly, they see this marked hearing loss in the patients as one of the most common features in the study. So we were interested in this further. I'll get back to that. Just say that here are some of the pathways that are in common to CSA and CSB proteins. They're involved in DNA repair, transcription coupled NER. They're involved in ribosomal RNA synthesis and transcription, chromatin remodeling, and then they commonly have these mitochondrial abnormalities. And we've recently found cross-species in worms, cell lines, mice, and human CS brain patient samples post-mortem, that there's a very severe mitochondrial phenotype in common cross-species, and we can improve that with NAD supplementation. And actually in mouse models, this was Morten's finding several years ago, that we could improve mitochondrial parameters by giving the mice NAD and a nicotinamide riboside. So we have been studying these pathways in Alzheimer's and in normal mouse models and in other diseases, and focused on some census, which I would like to discuss a little about here, because I think that we were trying some interventions in these particular using census. We've also studied the microbiome and just published a paper showing that the microbiome is severely changed in Alzheimer's mice, but dramatically improved after NAD supplementation. So it really improves and normalizes the microbiome spectrum. So I'll talk about some hearing studies that we've done, and this is cochlea, the inner ear, where the hearing occurs. This is a snail, and the sound goes through here in increasing frequencies. And in the cochlea from CSA and CSB mice, we reported a little while back that there was a lower level of NAD. And in the cochlea from the inner ear from the mouse models, both CSA and CSB. We then did an intervention study here. And in this case, it's only 10 days that we're giving NR in the drinking water. And we looked at the hearing, how it changed. We used ABR, which is a standard technique used to measure the hearing in humans as well. And you get these brainwave activities using this approach. And here is what happened at 5 weeks of age. You see that the CSB mice have less hearing at the 32 kilohertz level. And then after the next 10 days of giving NR, the CSB mouse has not lost any further hearing, whereas the one that's untreated has lost further hearing. So we completely blocked the progression of hearing loss in the CSB mice, both A and B genotypes. And so we've explored the mechanism here a good deal further. And I don't have time to talk about all the kinds of studies one can do in the inner ear. I'll focus on that we looked at these synaptic transmission in particular and found that there was a significant deficit in the synaptic transmission in the inner ear in the first synapse to the efferent nerve. And later we, in further studies, found that this may correlate with this ribeye protein that sits at that membrane, the synaptic membrane, and that is an NAD sensitive protein, has an NAD pocket. So this is also seen in the retina, and there are good results using NR as well in studies with retina, and that may be important in this context. So when we looked at various transcriptomics in the CS, we found that there was a change in synaptic transmission, particularly improved by NR. And so taking this a little further to a more normal situation, we've now been looking at age-related hearing loss, which is very commonly occurring. Of course, most of us suffer from age-related hearing loss, and it's very common in the population, so if anything can be done, it would of course be important. And so here we are looking at various mouse models, and interestingly, the type of hearing loss you see with aging is the sensorineural hearing loss. That's also the same mechanism that's seen in Copenhagen syndrome. So we can go on studying the same hearing deficit mechanism. And this work was done by Mustafa Okur, and it's not published yet, but ongoing here. We are looking at various mouse models. We're looking at various ages and various kinds of interventions. And importantly, C57 black has a cadherin mutation, so after a period of time, a normal C57 black mouse will lose its hearing anyway, so we have to either look at those early or use mouse models that don't have that kind of background. So we have an mTKIMA marker model that we've used for mitophagy marker on a mixed background, and then we have a CBA model which is widely used in hearing studies. And so this shows that we have lower levels in the inner ear of NAD, and after NR treatment they increase significantly with age here. And then we looked at this mTKIMA mouse and took it out from 2 month, 8 months to 12 month, and you can see there's a marked hearing deficit when you get to 12 months. That's improved dramatically by NR intervention. So here we use long-term NR in the drinking water, and we see a significant improvement of the age-related hearing loss. So it's a matter of how much hearing they lose with time. We're not restoring any hearing, but we're blocking the digression of the hearing. And when you look at these ABR waves, you can identify various pathways. And just to show you here that wave 1 in particular here is the red one is after NR. It is a broader amplitude than it is without NR. And this is likely ascribed again to a function of NR on the synaptic activity in the efferent neuron. We also identify various synaptic pathways with in various pathways analysis that we've done here in this model. And with a KEGG disease model, also clearly see synaptic pathways and also cardiomyopathy that's deficient in this model. We've looked more at specific pathway proteins, and some particular interesting ones are involved in oxidative damage responses. They are increased dramatically with age, and then they're improved after NR in these cases here. So this is we're doing much to try to understand the pathway more specifically here. We then also use the CBA model, and here we have to go out a long period of time before we see the improvement of NR, but we do see that here when we get out to 21 and 27 months of treatment. And we also did a study where we asked if we started NR intervention sort of at a later stage, would it still have an effect? And it shows that it does have an effect here. It was a 15-month, ongoing for a month and a half, and looking at individual mice, you can see there is a significant difference. So it's like in many other aging interventions, it's never too late to try to make improvement, and so here we can do it at a late stage. So these are the conclusions from this, and we're finishing this work, and I already mentioned all these things, I think, in talking through this. So we're also doing a hearing intervention in Alzheimer's mouse models, and we have developed a specifically interesting mouse model in Alzheimer's that's DNA repair deficient. And when it's DNA repair deficient, based on some findings originally in patient material, then you see more Alzheimer development, faster Alzheimer's, more severe symptoms, suggesting that DNA repair is protective of Alzheimer's. And also we see increased hearing loss early on when you deplete DNA repair in an Alzheimer's mouse compared to the Alzheimer's mouse that's normal, so to speak. And in Alzheimer's studies, we've done a number of things to look at function of NR, and again we see a very dramatic improvement of synaptic transmission, here using long-term potentiation with electrodes, electrophysiology, showing that the AD Paul beta mouse improves to normal levels when you treat them with NR. So we're very interested in how NR works at the mitochondria, how it works at the synaptic transmission. And I'll finish here and thank the people here, importantly Deb and Naz here who did this work, and Evandro who's now in Oslo, and Eugen now in Shanghai, participated a lot in the Alzheimer's mouse models here. And we appreciate these collaborations with the hearing colleagues and at Salk Institute and our colleagues here in Denmark as well, and the members of my group in Denmark and Lene. Thank you very much.
Thank you so much, Will. That was really a great talk. We have a question from here.
I noticed it wasn't a fantastic talk.
No, it was a fantastic talk.
Every other talk is fantastic, but thank you anyway.
We have had a fantastic program.
A great
It was
Thank you very much. Thank you.
Sorry.
Hi, really interesting talk. People have, there's been association with like the loss of dopaminergic signaling with hearing, and dopaminergic neurons is thought to be particularly, vulnerable in aging. Do you think that there's something going on with dopaminergic neurons in particular? I saw you had some dopamine synapse genes up there.
Yeah.
So.
No, we're not really been looking at dopamine. Glutamine transmission seems to be very important in Alzheimer's, and we haven't really looked at the dopamine levels. Maybe that's a good idea. Yeah, thanks.
Thank you. I was just reading the cocaine syndrome prenatally is usually diagnosed by taking the DNA recovery in chorionic villi and the
Resumption of
amniocytes. So I was wondering if it could be advantageous for the future research to try and treat those cells, the fetal cells, with NAD+ or NR.
Yeah, that is the hope that it could be. So you were talking about the resumption of RNA synthesis kind of assay?
Yeah.
Is that what it is? Yeah. We've tried to look at that, but we've gone a little further to look at other things that happen with CS. But it is clearly the hope that this could improve the situation for them. Yeah. I mean, we have cellular experiments, mouse experiments, nematode experiments, but we don't have human experiments. We don't really, it's very difficult to assemble these patients, and they're infrequent, and we don't even really know yet if they have lower levels of NAD in their blood or in their brain or anything. By the way, this goes into the brain, so it functions in the brain.
So I actually have two questions, one related to the first one and one to the second one. So related to the first one is because indeed these mouse models and genetic background really changes how the mice develop hearing loss.
Yeah.
Do you think that still NR would have the same effect on, or the phenotypes might be actually slightly different?
No, I mean, it's like all mouse work here. At least we have the two phenotypes. The mice don't have a very strong phenotype, so they are not excellent models for Cockayne syndrome, but they do have this hearing loss that's seen in patients. So the mouse is not useful for all kinds of studies, but we think it's useful in the case of hearing loss. But whether that really helps the patients needs to be done, obviously. So it's like, that's what we could say, it seems to.
And about NR supplementation, could you try to make something a little bit more local to then really show that it's the local amount of NR that would make a difference and not maybe something that comes from?
Yeah, I mean, by the way, we've tried other things. We've used NMN, we've used other compounds that stimulate NAD supplementation. But in this work, we are looking locally at the cochlea. I showed that we are looking at the cochlea. We see the increase in the cochlea. In the brain, when we do the Alzheimer's work, we look in the brain at different brain regions. And then we look at supplementation and see what changes in the brain or in the region. Yeah, they're fed orally, yeah, because it's, for a number of reasons, easier to do. But incidentally, we now think through this work with the Alzheimer's model that there might be a different level of damage to DNA or DNA damage signaling in the cochlea compared to the cortex. So that maybe one problem related to hearing is that it's a sink for DNA damage. So various DNA damage pathway signaling is upregulated in the cornea, more H2AX formation and stuff. And that's kind of unusual in DNA repair, where we're used to thinking that there's the same level of damage for everything everywhere. Like for UV, that's the case. But for these things, it may turn out there are big differences in the induction of DNA damage in different regions of the genome due to exposure.
Steve?
Could you briefly comment on what is better, NR supplementation or NMN?
So this is really being widely discussed at meetings that I go to, and there are two different camps, as you well know. And I would put it this way, that in our studies, NMN is hard to come by in larger quantities. So for the mouse models, we've usually used NR, but for cells and nematodes and organisms, we've used both. When we look at the features that we're interested in, we see the same results with NR and NMN. So I know there's an endless discussion about pathway differences, and that is true, but that's the same in our hands. Now, in addition, we have so much safety work done with NR at this point that we, I think, know that there are very few side effects. A few were mentioned at this meeting, but very limited, a lot of human studies done. With NMN, there are different compounds being made by different companies at this time, and we don't really, there's one clinical study that suggests possibly no side effects, but much less than with NR. And so different compounds and less studies makes me think that for something that could be applied in humans readily, you would prefer NR.
And do you take NR supplements yourself?
Occasionally I do, yeah. I mean, I know many people that do. But yeah, I mean, it doesn't, you know, it doesn't show, several studies that have been done in human studies, we heard also here, don't show any particular benefits on short term. So I don't know, I mean, but it's possible that in some conditions, when NAD is lower, then you will have a big effect. Or that in long-term prevention. But I'm not paid by any company, so I'm just speaking from my experience, and I get the drugs for free, but I don't get any money from any of the companies.
Quick question from Anna.
I would like to ask your opinion, because I'm now adding the third camp here, because I'm coming from the clinical side and not the company side. We have used niacin in a human study in 2020, and we showed that it's a very efficient niacin, nicotinic acid, for patients with mitochondrial myopathies and controls. And in 10 months of treatment, there's a considerable increase of mitochondrial biogenesis in the muscle, and I mean, there's in controls and in the patients, so the patients also benefited like remarkably of because we knew that there is an NAD deficiency, like absolute NAD deficiency in them. So I think it is, I mean, niacin has not much been mentioned in this study, and I would just like to, in this meeting, and I would like to hear your
It's a good time to mention it. Yeah, it's not the mitochondrial study with the mitochondrial eye problems. No, okay. Okay.
Great point. All right, thank you so much, Will.
Thanks.
Fantastic talk. Really appreciate it. Thank you so much for being here, finally.